Hello everyone. Today let us study about the topic viral diseases in plants under the main subject agrochemical and pest control. Viruses are submicroscopic entities capable of causing disease. They are a piece of nucleic acid surrounded by a protein coat. Once inside the plant cell, the nucleic acid portion directs the plant cell to produce more virus nucleic acid and virus protein, disrupting the normal activity of the cell. Viruses also cause many important plant diseases and are responsible for huge losses in crop production and quality in all parts of the world. Infected plants may show a range of symptoms depending on the disease, but often there is a leaf yellowing either of the whole leaf or in a pattern of stripes or blotches. Leaf distortion, example curling or other growth distortions, example stunting of the whole plant, abnormalities in flower or fruit formation. In general, viruses are seldom lethal to plants, but they do severely affect the host both in quantity, quality and longevity. Symptoms may often be very characteristic for a specific virus on a specific host. Symptoms along with other criteria are used to identify virus diseases. An advanced array of symptoms can be recognized today as expressions of viral diseases in plants. Hence, in this episode, the following aspects are going to be seen: biology and morphology of virus, transmission of plant viruses, survival and dissemination, plant virus interactions and symptoms of plant viruses, and finally, management of plant virus diseases. Let us study about these aspects one by one. The first aspect, biology and morphology of virus. Biology of virus. Viruses are minute parasitic organisms too small to be seen by a light microscope. They consist only of a nucleic acid core and a protein coat and are unable to grow outside the living cells of their plant hosts. They cause a bewildering variety of symptoms, some of which are very hard to distinguish from mineral deficiency or herbicide damage. Virus infection can reduce yields drastically but rarely kills the plant. Viruses have no means of movement and rely on other organisms to transmit them from disease to healthy plants. These are often sap-sucking insects such as aphids, thrips, whiteflies, or by leaf-feeding beetles or plant-feeding mites. Soil-inhabiting nematodes and fungal pathogens can also transmit viruses. Some viruses are also very contagious and can be transmitted on pruning knives or gardener's hands. Viruses are also transmitted from one plant generation to the next via infected cuttings or graft material, and a relatively small number can pass through infected seed. Once a virus particle enters the host plant cell, it hijacks the cell's synthetic chemistry to create more virus particles, diverting the cell's resources from normal activities. The virus nucleic acid takes over the cell's own nucleic acid, instructing it to synthesize new virus nucleic acid and protein. In the process, the plant's normal growth activities are disrupted and the visible symptoms are produced. Morphology of virus. There is a simple structural principle that applies to virtually all viruses in their mature form. Virus particles are composed of two principal parts: the genome, that is made of nucleic acid, and a protective shell that is made of protein. The protein shells of plant viruses are assembled in according with one of the two fundamental types of symmetry. The first type of virion is helical. The elongated viruses come in two major variants, rigid rods and flexuous filaments. In both of these variants, the nucleic acid is highly ordered. It assumes the same helical conformation as the proteinaceous capsid. The second type of virus particle is icosahedral. The variations of this basic shape include bacilliform virions and twin virions, which are composed of two joined incomplete icosahedra. In the icosahedral virions, the genomic nucleic acid forms a partially ordered ball inside the proteinaceous capsid. The icosahedral and elongated virions alike can self-assemble in a test tube if the nucleic acid and protein subunits are incubated under proper conditions. Moving on to the second aspect, that is transmission of plant viruses. Plant cells are eukaryotic cells that are similar to animal cells. Plant cells, however, have a cell wall that is nearly impossible for viruses to breach in order to cause infection. As a result, plant viruses are typically spread by two common mechanisms, that is horizontal transmission and vertical transmission. Let us study about these two mechanisms briefly. Horizontal transmission. In this type of transmission, the plant virus is transmitted as a result of an external source. In order to invade the plant, the virus must penetrate the plant's outer protective layer. Plants that have been damaged by the weather, pruning or vectors such as bacteria, fungi and insects are typically more susceptible to a virus. Horizontal transmission also occurs by certain artificial methods of vegetative reproduction typically employed by horticulturists and farmers. Plant cuttings and grafting are common modes by which plant viruses may be transmitted. The second mode of transmission is vertical transmission. In vertical transmission, the virus is inherited from a parent. This type of transmission occurs in both asexual and sexual reproduction. In asexual reproductive methods such as vegetative propagation, the offspring develop from and are genetically identical to a single plant. When the new plants develop from the stems, roots, bulbs, etc. of the parent plant, the virus is passed along to the developing plants. In sexual reproduction, viral transmission occurs as a result of seed infection. Moving on to the third aspect, that is survival and dissemination. Only relatively few viruses such as tobacco mosaic virus or TMV rely on long-term survival in the environment and on passive mechanical transmission from plant to plant. Most plant viruses are actively transmitted from infected to healthy plants by a living organism called a vector. Plant feeding arthropods, nematodes and plant parasitic fungi are the major types of vector organisms for plant viruses. Among these, aphids and whiteflies are capable of transmitting the largest number of virus species. Most viruses are actively transmitted to healthy plants in a matter of seconds, hours or days by vectors. In addition, some viruses are transmitted from infected plants when these plants are vegetative propagated, for example, through tubers or vertically through seeds or pollen from infected plants. This type of transmission is particularly important for virus survival during the winter. Transmission of viruses by a vector is a very specific process. Each particular virus can be transmitted by only one vector type, example aphid, and not by another. example whitefly. Conversely, each vector species, example green peach aphid, Myzus persicae, can transmit some viruses, example beet yellows virus, but not others, for example citrus tristeza virus, despite the fact that these viruses are genetically very similar to each other. The interaction between a virus and its specific vector that results in virus transmission varies from different virus vectors. The relationships that have developed between viruses and their vectors are complex and of considerable interest to plant virologists because vectors provide the main method of spread for many viruses that cause severe economic losses. For some virus combinations, the interaction between a virus and its vector is very superficial and it is the result of attachment to the external surfaces of the vector mouthparts. It takes at least 12 hours for these viruses to circulate in their aphid vector before they can be transmitted to another plant. For other plant virus vectors, the relationship is very intimate and the viruses actually multiply in the cells of their insect vectors. Moving on to the fourth aspect, that is plant virus interactions and symptoms of plant viruses. Let us study about them one by one briefly. Plant virus interactions. Viruses are capable of infecting virtually all species of cultivated and wild plants. However, host ranges of individual viruses vary from very narrow to very broad. For instance, citrus tristeza virus infects only a few species in the citrus genus, whereas cucumber mosaic virus infects over 1000 species in 85 plant families. Susceptibility or resistance of plant species and cultivars to viruses is determined primarily by the plant genotype. Plants possess active and passive means of preventing virus infection. Passive defenses are due to the failure of the plant to produce one or more host factors required for virus reproduction and spread within the host. Active defenses include detection and destruction of the virus infected cells due to the function of specific resistance genes in the plant. Normally, resistance genes are active only against a particular virus. In addition, plants possess a general defense system that is somewhat analogous to the animal immune system. The major difference between the two is that the immune system in animals targets a pathogen's proteins, whereas the plant defense system, which is called RNA silencing or ribonucleic acid silencing, detects and degrades viral RNAs. Depending on the particular combination of virus and host and on environmental conditions, a plant's response to infection may range from a symptomless condition to severe disease and plant death. In some cases, small necrotic or chlorotic spots called local lesions develop at the site of infection. In most cases, viruses spread throughout the whole plant and cause a systemic infection. The end result of virus infection is a reduction in plant growth, lower yield, inferior product quality and economic loss to individuals who work in the plant industry. Most of the symptoms induced by viruses can also occur due to adverse environmental conditions or diseases caused by other plant pathogens. Because of this, correct diagnosis of viral diseases normally requires laboratory test. Let us study about the symptoms of plant viruses. Symptoms are the expression of the diseased condition of the plant. Almost all virus diseases seems to cause some degree of reduction in yield, and the length of life of virus infected plants is usually shortened. The most obvious symptoms of virus infected plants are usually those appearing on the leaves. but some viruses may cause striking symptoms on the stem, fruits and roots. Let us study about the symptoms of virus on plants. The symptoms may be external or internal, external or macroscopic symptom. They may be local or systemic. These are the symptoms produced at the site of artificial inoculation on leaves with virus. Some of the local symptoms are chlorotic local lesions. Infected cell lose chlorophylls and other pigments. For example, tobacco mosaic virus on cowpea host. The next is necrotic local lesions. The infected cell die. Example, tobacco mosaic virus on Nicotina glutinosa host. The next is ring spot local lesions. This consists of central group of dead cells near inoculated area which forms a necrotic ring. For example, potato virus on Chenopodium amaranticolor host. The next form of symptom is systemic symptom. In almost all viruses of plant occurring in field, the virus is present throughout the plant. The symptoms may be variegation, malformation and other types. Variegation, which is also called as color breaking. Decoloration in patches on the leaves occur, sharpness of boundaries can be seen, distribution and discoloration of lamina can be observed. Under variegation, we can see mosaic, line pattern and mottling. Mosaic. Mosaic are characterized by light green yellow or white areas intermingled with normal green of leaves of fruit or of whitish areas which are intermingled with the areas of the normal color of flowers or fruits depending upon intensity or patterns of discoloration. Mosaic type symptoms may be described as follows. They may be streak or stripe, for example, red stripe of Jover. They may show vein clearing, for example, BCMV virus. They may show vein banding, example, beet curly top. Internal venal mosaic light discoloration restricted in between the veins can also be observed. The next is line pattern. Under this, we have oak leaf pattern or OLP, for example, apple oak leaf. Systemic rings AMV on tobacco. The next is mottling. If the discolored patch of a variegated leaves are rounded, the variegation is usually designated as mottling. The next is malformation. Change in leaf form, any deviation from normal. This includes uneven growth of leaf lamina, leaves become curled, brittle and show prominences and depressions, upward and downward curling, vein distortion, leaf annation, galls and tumors. The next important form of symptom is based on internal or microscopic. These include inclusion bodies. Inclusions are microscopic bodies produced by some of the viruses. These are produced in cytoplasm or nucleic acid. Cytoplasm. Amorphous or amoeboid, also called as X bodies, produced by spherical or oval viruses. Example, CMV. Next is crystalline. These types of inclusions are produced by rod-shaped viruses. Example, cactus virus and petunia ring spot. Nucleus. Example, tobacco H virus, rectangular plates, BCMV strain, Pisum virus 2 and isometric crystals. Moving on to the last aspect, that is management of plant virus diseases. Although there are virtually no antiviral compounds available to cure plants with viral diseases, efficient control measures can greatly mitigate or prevent disease from occurring. Virus identification is a mandatory first step in the management of a disease caused by virus. The subsequent strategy for management will depend on the means by which a particular virus enters a crop, how the virus is transmitted between plants within a crop, and how the virus survives when the crop is not being grown. Preventative measures may include use of certified virus-free seed or vegetative stocks, elimination of the virus reservoirs in the surrounding wild vegetation, and modification of planting and harvesting practices. If the virus is known to be transmitted by a particular vector, control or avoidance of this vector is of paramount importance. For instance, insect, nematode or fungal vectors can be controlled by insecticides, nematicides or fungicides respectively. An alternative strategy for virus control is utilization of natural or engineered resistance to virus infection. If available, natural virus resistance genes can be introduced into crop cultivars by conventional breeding. These natural resistance genes are often found among the many cultivars that are available for a particular crop, or the resistance genes are found in a related plant that have been identified near the center of origin for that crop plant. Genetic engineering, the transfer of genes between specific organisms using enzymes and laboratory techniques rather than biological hybridization, allows cross-species introduction of such genes. Furthermore, virus resistance can be designed by presetting the plant ribonucleic acid silencing defense system. This is achieved by introduction of fragments of viral nucleic acid into the plant chromosomes. Such transgenic resistance confers immunity to infection by the virus from which the nucleic acid was derived. The effectiveness of this type of transgenic resistance is illustrated by the successful management of papaya ring spot virus in Hawaii. Although genetic engineering offers unlimited opportunities for generating virus resistant crop plants, its large scale application has met some resistance from researchers, supervising agencies and the public. A better understanding of the potential risks associated with introduction of genetically modified plants into the environment is needed to help assess the safety of plants that have been genetically engineered for virus resistance. Finally, to conclude with today's topic, plant viruses and viroids are diverse and unusual group of plant pathogens that infect and cause disease in many crop plants. Because these pathogens depend on the normal cellular machinery of the plant host for reproduction, it is difficult to eliminate them without damaging the host plant. Therefore, most management strategies for diseases caused by plant viruses and viroids are directed at preventing infection of the plant. Thank you.